Electrophoresis was first described by Tiselius in 1937. It is a process by which charged particles move through a medium in the presence of an electric field at a given pH. The charged particles move at constant velocity during movement. The electrical force acting on the particle is equal to frictional force on the viscous track, where equal to Fv is the strength of electric field volt per centimeter and Q is the net charge applied on the particle. This is the V equal to velocity equal to Eq upon F, where mu equal to V upon E and the mu is the EQ by is EQ upon F into E and obtained is mu equal to Q upon F. Under the influence of electric field, these charged particles will either move toward cathode or anode depending upon their charge. The phenomena is called electrophoretic mobility. Electrophoretic mobility denoted by mu, and the electrophoretic mobility is the ratio of the velocity of the particle to the field strength. So when electric field is applied, the charged molecule will move and separate according to their electrophoretic mobility. In this way, from a mixture of charged molecules, we can separate each molecule in the form of a band. The instrumentation is of the electrophoresis is the electrophoresis equipment consists of two items: electrophoretic tank and electrophoretic power supply. The electrophoretic is of two types: vertical electrophoretic tank and horizontal electrophoretic tank. A horizontal used tank used for agarose gel electrophoresis. The two main movement types, that is phase polyacrylamide gel electrophoresis. The polyelectrophoresis phase is called as polyacrylamide gel electrophoresis, and the SDS phase is performed in vertical gel electrophoresis system. The polyacrylamide gel is formed between two glass plates which are held together by clamp, and they are separated by plastic spacer. The size of the gel is usually 12 into 14 centimeter and the thickness of gel is 0.5 to 1.0 millimeter. A plastic comb is placed in acrylamide solution and it is removed after polymerization of gel. It creates well inside the cell in which protein samples are loaded. The plastic comb, this is the polyacrylamide gel electrophoresis tank, contains plastic comb, stacking gel and resolving gel, glass plates and plastic space. For the separation of protein samples, two different gels are prepared in the same glass plates. The upper gel, which contains the well, it is where proteins are loaded, it is called stacking gel, and it is made up of 5% acrylamide. The lower gel which separates the protein is called as the lower gel which separates the protein is called as resolving gel, and it is made up of 10 to 15% acrylamide. The purpose of stacking gel is to concentrate the protein sample so that sharp protein bands are absorbed. During electrophoresis, when the tracking die reaches to the bottom of gel, the current is switched off, the gel is removed, and it is stained with a protein staining dye, commissive brilliant blue, for a few hours. The gel is then washed with a de-staining solution containing glacial acetic acid methanol in water. After de-staining, the polyacrylamide gel appears transparent and the protein bands appear dark blue in color. The vertical gel electrophoresis system contains upper anode and cathode electron charge in the upper butter chamber and gel and lower butter chamber. This is the vertical gel electrophoresis. The applications of page is polyacrylamide gel electrophoresis is the it is used for the separation of plant, animal and microbial proteins. It is used to calculate molecular weight of a protein. It is done by running protein sample markers with the unknown protein. This technique is used to determine purity of a protein sample. It is used in Western blotting. And the next topic is agarose gel electrophoresis. Agarose is a linear polysaccharide molecular weight containing 12,000 and it is made up of repeating units of a disaccharide agarose biose which is made up of galactose and 3,6 anhydrogalactose. Agarose is a compound which is present in agar which is obtained from seaweeds in agarose gel electrophoresis. Agarose at a concentration of 1 to 3% in suitable buffer is used. Agarose gels are performed by suspending dry agarose in a suitable buffer such as trees, EDTA buffer, and then the solution is boiled until a clear solution is obtained. The solution is powered in a gel caster and the solution is allowed to cool at low temperature. When a plastic comb is inserted in the agarose solution so that wells are performed, when the agarose solution cools down, it forms a solid gel. The solid gel has cross-linked structure which forms pores inside the gel. The pore size is controlled by concentration of agarose. Concentration of agarose. If concentration is high, then pores are very small. If the concentration is high, the pores is small. This is the diagrammatic representation of the agarose gel electrophoresis. This is the well, and here the band represent blotting techniques of the agarose gel electrophoresis. There is a band and where gel presents. This is the platform pattern of agarose gel, and this is the anode. Here gel present and containing positive cathode and negative anode charge. This is the horizon pattern of the agarose gel electrophoresis. If in this system the gel is placed on a platform, the gel is covered with a buffer solution. The electrophoresis system is connected to power supply which provides electric current to the electrophoresis system through cathode and anode. The buffer is required to maintain ionized state of nucleic acid to be separated. DNA and RNA are negatively charged due to presence of phosphate group and they move toward the cathode. The separation of DNA molecules occurs due to different mobility of DNA molecule due to their size. Small DNA molecules can move freely inside the gel and large molecules move with difficulty. Since DNA molecules separate according to their size, molecular weight of DNA can be calculated by DNA molecular weight markers. Before electrophoresis, the DNA sample is loaded in the well. The DNA sample contains sucrose or glycerol, which provides density to DNA solution so that it enters in the well. DNA sample also contains a tracking dye, that is the bromophenol blue, which helps to monitor the run of DNA during electrophoresis. The size of agarose gel is 25 into 12 cm and it is run at a voltage gradient of 1.5 volt per centimeter. The voltage gradient is 1.5 volt per centimeter. After electrophoresis, the DNA is stained with fluorescent dye, ethidium bromide, and it is observed in ultraviolet light, that is UV light, 300 nanometer. Ethidium bromide binds between the nucleotide bases and shows orange-red fluorescence, and so DNA bands appear orange in color.